Hello everyone, welcome back to the next lecture on molecular luminescence spectroscopy in this MOOCs course on advanced analytical technique, and the topic of today's discussion is the instrumentation of molecular luminescence spectroscopy. The important components of a molecular luminescence spectrophotometer are the light source, the excitation monochromator, the sample holder or sample compartment, then emission monochromator because we have two different light radiations. We have an incident light which is absorbed causing excitation. So the monochromator through which it is passed, it's called as excitation monochromator. On the other hand, when the electrons comes back from excited state to ground state and they emit visible light radiation called as fluorescence light, then also we placed at that point also we placed a monochromator to select some wavelength while excludes the other, and that monochromator is called as emission monochromator. Then we have the detector to detect the signal, the intensity of emitted light, and finally signal processing unit and output system. Signal processing, we have amplifier to amplify the signal, and then we have computer to display the results in the form of a spectrum. The basic design of a fluorescence spectrophotometer or spectrofluorometer. These fluorescence spectrophotometers are also known as spectrofluorometers. Their basic design is quite similar to our UV visible spectrophotometers, because UV visible spectrophotometers also have light source, then we have sample tube, we have detector, we have monochromator also. We have one monochromator in case of UV visible because we only place it before the sample tube so that incident light passes through monochromator, few selective wavelength, and then it enters into the sample tube. But in this case, since we have incident light radiation and we have emitted light radiation as well, so we use two monochromator. We have one photomultiplier tube as a detector in UV visible. Same is the case with spectrofluorometer. We have photomultiplier tube as detector, but for multiple analysis we can use SSD, the solid state array detectors based on charge coupled devices that we have discussed in case of ICP, inductively coupled plasma. So the design is quite similar to UV visible spectrometers, although in some of these spectrofluorometers we have extra features for fluorescence. For observing fluorescence, we keep the sample at low temperature to properly observe the phenomena of fluorescence, and maintaining that low temperature is possible by cooling the sample tube with liquid nitrogen, and for liquid nitrogen the temperature is around -196 degree Celsius, and this is only possible by Dewar flask. I hope you remember that we have discussed it when we were discussing about ICP, and I told you that the large volume of argon gas at high flow rates are required for ICP because it is used for three purposes: as a coolant, as an auxiliary gas for creating plasma, and for creating aerosol. And that much flow rate can be maintained only by using liquid argon gas, and liquid argon gas is stored in the Dewar flask, which is a compartment of two outer linings made of steel and has a vacuum which avoids heat transfer due to convection and conduction, while there is a coating of silver to avoid any radiant heat, and then there is a coating of silver to reflect any heat from outside coming into the flask. So in ICP we use this Dewar flask for argon gas storage in liquid form. Here in this case of phosphorescence, if our spectrofluorometer has a provision for fluorescence analysis as well, so for that particular case, in order to observe it properly, we have arrangement to keep the sample tube at very low temperature by liquid nitrogen gas, because at low temperature the delay between excitation and emission can be observed. Otherwise, if we are having this phenomena of phosphorescence at room temperature, the delay cannot be observed because it's very quick. So if we want to study that delay, we have to carry out that particular process at very low temperature. This is a schematic diagram of a spectrophotometer where we have a light source which produces light radiation that passes through an excitation filter or a monochromator selective wavelength and passes through the sample. When these wavelength passes through the sample, some of the light is absorbed due to the interaction with the molecules of the sample, while the remaining light is transmitted, of course, in a straight line. So if the intensity of light radiation that is coming in is I naught, the intensity of transmitted light is I, and the intensity of light that is being absorbed for causing excitation is I naught minus I, or maybe P naught minus P. So since transmitted light goes straight, undeviated, and we are not concerned with this transmitted light, the detector is not placed here, rather the detector is placed at 90° here, but before the detector we placed an emission filter or emission monochromator, because when fluorescence is taking place it emits light in all direction, means fluorescence is emitting light in all directions. We have light radiations in all direction. We capture light in the perpendicular direction. We put a monochromator or emission filter here that selects few wavelengths while excluding the other. These radiation passes through it and finally falls on the photomultiplier tube, which has a photocathode. We have a cathode over here. In between we have diodes. We have already discussed it in atomic absorption spectroscopy. When photons falls on it, electrons are ejected, and when electrons reaches to the anode, current is generated, an electrical signal is created. Finally, this signal is amplified with the help of an amplifier, and output is shown in the computer. Sometimes we also use a reference multiplier if we are having some reference sample as well. This slide explains the previous schematic diagram. The yellow circular button, this one is the light source. Then the light radiation that is being generated by the yellow colored light source passes through the monochromator or excitation filter, passes through the monochromator and then falls on the sample that is of green color. There will be some interaction between the sample and the light radiation. The transmitted light, of course of lower intensity, passes, while there will be interaction, there will be some absorption causing excitation of the electrons. Then we again have a monochromator which selects some light radiations of our interest while excluding the other. Then these light radiations falls on the detector, which is a photomultiplier tube. That signal of the photomultiplier tube is amplified. A small downward pointing triangle is also present here. It is positioned centrally at the bottom, possibly indicating direction, grounding or flow. It is just showing that light radiation at the end is going towards the lower side, where we have an amplifier device which amplifies the signal. The small signal is converted into a big signal that can be readable, and that readable signal is then presented on the output device. So as I said earlier, so here this slide shows that the sample tube which is used for placing the sample, it is made of glass or quartz, but most of the time a glass sample tube or glass cuvette is of course of low cost. It is quite cheaper as compared to quartz, but its optical property or transparency is lesser than quartz, and it is not very useful if the temperature is high. Quartz is more durable, but it is costlier. So for research purpose we always use quartz tube, but if we are explaining this phenomena to undergraduate students, and we are allowing undergraduate students to do the analysis on a spectrophotometer. We give them glass cuvettes because they sometimes broke, because they sometimes uh, not very carefully using these cuvettes and resulting into the damage of these cuvettes, and if quartz cuvette gets damaged, it is very costly. And further, for their learning and experimentation as an undergraduate student, glass cuvettes work well. While for research purposes, for more reliable data, for very high precision and accuracy, we always use quartz cuvettes. And the detector, as I said earlier, is placed at right angle to capture fluorescent emissions only, because we do not want to capture the transmitted light. Even, and if the transmitted light is also captured by the detector, we'll get some extra signals which will cause complications in the spectral studies. So we always put the detector at right angle to get only fluorescence emissions, and to have a reliable data. This is again a diagram where we have a light source. Xenon lamps are quite common for fluorescence analysis, while in case of UV visible spectroscopy, if we are using UV radiations, we use hydrogen lamp. We sometimes also use deuterium lamp. For visible region, we use tungsten filament lamp. While in this particular case of fluorescence or spectrophotometers, we use xenon lamps. Xenon lamps covers the UV as well as visible region, and these xenon lamps produce bright white light. Remember one thing, fluorescence is a cold body phenomena, means the sample is not heated to emit light radiation as in case of emission. This is the difference between emission spectroscopy and fluorescence spectroscopy. In case of emission spectroscopy, the sample is excited by heat, either by direct aspiration, direct flame heating, or by electrothermal heating, or by plasma, so that the electrons of the molecule, so that the electrons of the sample get excited to a higher level. When they come back, they emit light radiation. Yeah. So in that case, it is a hotter body emission. But in case of fluorescence, it is a cold body emission, means the sample is not heated, the sample electrons are excited to the next level by absorption of light energy. So there is no change in the temperature of the body. So we have a xenon lamp, then excitation monochromator, the light is coming, passing through the sample. Then we again have a lens and a monochromator placed at 90°, and then we finally have a detector over here to detect the signal and present those results in the form of a spectrum. So the working principle of fluorescence spectroscopy, it is an analytical technique that measures the concentration of an light in a sample by measuring the amount of light being emitted at a characteristic wavelength upon absorption of light of requisite energy. Or in other words, I can say that according to the principle of fluorescence spectroscopy, the concentration of an analyte in a sample is directly proportional to the intensity of light emitted upon absorbing light of requisite energy. So, if the intensity of light that is being emitted is high, this means that the concentration of the sample is high. So, if the intensity of light that is being emitted is low, the concentration of analyte in the sample is low. While on the other hand, if the intensity of light emitted is high, this means that the concentration of analyte in the sample is high. So, there is a direct relation, and this serves as the principle of fluorescence spectroscopy. And the light emitted, it is always of longer wavelength. The light emitted has a longer wavelength than the light which is absorbed. Means longer wavelength means lower energy than the light which is absorbed. So, light which is absorbed has high energy and low wavelength. And once again, I would like to stress upon the fact that unlike emission spectroscopy, here sample is not heated, no heating is involved. The light emitted in fluorescence spectroscopy is perfectly a cold light or a cold body radiation, where the sample or the molecule under study is not heated. It is the light without being heated. It is the light without the sample is being heated. Again, this is an actual image of a fluorometer. The size and appearance looks like a UV visible spectrophotometer. They are quite similar. But in UV spectrophotometer, absorption is taking place, while in this case both absorption and emission are taking place. This is lamp. The light created by the sample, there are filters placed in the path of the light that select few light radiations, then it falls on the sample, and it is not in the straight line, as I said, because in a straight line we have transmitted light. So there is another filter placed at 90 degree called as the second monochromator or emission monochromator, and finally this light falls on the photomultiplier tube. These fluorometers are single beam as well as double beam. Single beam has one slot for the sample only. Single beams have one slot for the cuvette. They have one slot. In first case we put the sample, in first case we put the reference here, allow the light radiation to pass through it, and fluorescence spectrized and a fluorescence spectrum is generated. Then in the second case we again open the cover of the machine, remove the cuvette, wash it, then fill it with sample, and again we put the sample, and then we put the sample inside, close the cover of the machine, and then allowed the light radiation to pass through it to create another spectrum for the sample. And in this way we compare the two spectrum, the spectrum for reference as well as sample, and if they are overlapping, if they are superimposing each other, then we can say that the sample is actually the reference material, while if the spectrum are different, we can say that the sample does not contain that particular molecule which is present in the reference material. So, if we are going for a low cost spectrophotometer, it is a single beam spectrophotometer, and a single beam spectrophotometer has a single light radiation that passes through one sample tube only. There is a slot for placing one cuvette at a time. First we use the same cuvette for analyzing the reference material, then in the second case we remove the cuvette, throw out the reference material, wash the cuvette, put the sample again, and then place it, and then place the cuvette again in its slot. Then we perform the analysis again. Then in the second run only we capture the sample details, and the two are matched for analysis. While in case of a double beam spectrophotometer, we have two slots, one for reference other for sample. They are little bit costly, but they makes our analysis quite easy because there are two different slots. In one slot we put the reference material, while in other slot we have another cuvette, we put the sample, we start the machine. A light radiation is created from the light source. For example, this is the xenon lamp. It creates a light radiation that passes through a filter, and then this light radiation is divided into the light radiation that is being created by the light source. It goes straight, and then there is a prism which divides these light radiations, which divide this light radiation into two different light radiations of equal intensity. Then we have mirrors, then we have focus mirror that turns this light radiation at 90 degree. Finally we have two slots, one for the reference cuvette, other for the sample cuvette. After passing through the sample and reference, we again have mirrors for focusing. These light radiations again passes, and then we have a detector which is placed at, and then we have a detector to detect the signal, and a spectrum is created. If these two spectrums have same peaks at same positions, the two spectrums are superimposable. We can say that the reference and sample are same, but if the peaks in the spectrum are different, the two spectra are not superimposable, then we can say that of course they are two different samples. This diagram shows you how light is being reflected, or it is diffracted to reach the sample in the machine. So xenon lamps creates a light radiation that passes through a filter. Then there is a monochromator. This is called as the emission. This is called as the excitation monochromator, and you all know, because we have already discussed, that monochromator consists of two parts. We have slits. We have two slits, entrance and exit slit. And we have grating or prism. Like in this case I have shown you a prism, so we can have prism or grating. In this particular example, this is entrance slit, then this light passes through this slit. Some of the light radiation are then reflected through this mirror, falls on the grating, again they are reflected, and finally focused through this exit slit. Then this is a lens, passes through, it falls on the sample, where some interaction between the light radiation and sample. The sample molecule absorbs light, the electrons are excited, they are going up to the excited state. When they return, they again emit light radiation, while the transmitted light goes straight, and the light emitted, it is emitted in all directions. So we capture the light emitted is emitted in all directions. We capture this light which is going at 90 degree. We again put a focus mirror that reflect this light radiation again to the second monochromator, which is the monochromator for emission. This is our emission monochromator. This one is our excitation monochromator. Then we have then the light radiations passes through entrance slit, falls on the mirror, reflected to the grating. This grating disperses light radiation into narrow wavelengths. It narrow down the wavelength of the light radiation. This narrowed down light radiation pass again reflected by a mirror, passes through, focus through this exit slit, and then focus through this exit slit, and finally reaches to the detector, where we have a photosensitive cathode which produces electrons, and these electrons are reflected consecutively by dinodes, and finally reaches to anode, creating an electric signal. Electric current is created. And spectra, and creating an electric signal or electric current, and spectrum is formed on the computer. So these are various steps that I have just discussed. Light from the light source is coming, that passes first of all through a monochromator called as excitation monochromator, and then which is having an entrance slit, then focus mirror, grating, again a mirror, and then exit slit. Then that light passes through the sample, causing fluorescence to occur, and the fluorescence light that is being emitted is captured via a mirror placed at 90 degree, at which reflects the light again at a specific angle, so that this fluorescence light is directed to the second monochromator called as emission monochromator. It passes through the entrance slit. Again there is a focus mirror which reflects light to the grating, which disperse light into individual narrow wavelengths. Then these narrowed down wavelength again reflected by a mirror and focus through exit slit finally falls on the photomultiplier tube, and a signal is obtained which is represented in the form of a peak on the output device. With this, I am going to end this lecture. Thank you very much. So we have learned about the instrumentation of spectrophotometer. In the next lecture, we will discuss about the components, means the light source, what kind of light sources are used. Then we will discuss about monochromators and also about the detector. Thank you very much.